What's up, Tumnad Lectures? In this video, we are looking at what we call the summary of gastric secretions. If you find it helpful at the end of it all, please make sure you like the video, comment down in the comment section, and then subscribe. Let's go ahead and get started, okay? So now, in the previous videos, we have been looking in detail of what we call the gastric secretions. Today, we want to look at what we call the summary of the phases of gastric secretions. We said that the gastric secretions are going to be having a lot of the components, the likes of the bicarbonate, the hydrochloric acid, the gastric lipase, the mucus, and the intrinsic factor. A lot of the substances they are making what we call the gastric secretions. The gastric secretions, they are going to be produced in 3 phases. What are the 3 phases? The first one is cephalic, the second one is the gastric, and then the last one is what we call the intestinal phase of gastric secretion. Now, let's have a summary of each one of them about the key important areas on each of these phases. The first phase we said is what we call the cephalic phase, and this phase is accounting for only 1/3 of the gastric juice within the stomach. And something which is very unique with this phase is that there is no food in the stomach. The stomach is producing a lot of the gastric juice, but there is no food within the stomach, meaning that this kind of the phase is more of the preparatory phase, okay? And the main cranial nerve, which is going to be driving all the processes leading to the production of the gastric juice during cephalic phase. You know how this cranial nerve is called? This cranial nerve is what we call the vagus nerve, cranial nerve number 10. It is such another a beautiful nerve. So now, what are the main stimuli of this cephalic phase to produce gastric juice during this cephalic phase? The first stimuli is going to be the smell of food, okay? And then the second one is what we call sight of food, just mere seeing the food. And then the third one is what we call taste of food, just mere tasting the food. The fourth one is what we call thinking about food. And then the last one, which is somewhere a bit minor, but it's also significant, is also hearing a frying sound, okay? What happens is that all these kind of the stimuli are going to be forwarding the information to the different cortex. For example, when you see the food, you're going to be having the information hitting the retina. From there, the information will be taken to what we call the visual cortex in the different areas of the cerebrum. From there, it will give off the information to what we call to the hypothalamus, specifically to the parasympathetic portion of what we call the hypothalamus. From there, it will give off descending axons to what we call to the medulla oblongata. In the medulla oblongata, we have what we call the dorsal vagus nucleus or dorsal nucleus vagus, which will give off the motor fibers of the vagus nerve that will come out through the jugular filament, and then it enters into what we call the abdominal cavity, and then it releases acetylcholine on different cells within what we call the gastric gland. Okay, so now all of these kind of the stimuli are going to be following the same pathway. For example, when you taste food, we're going to be having sensory fibers of cranial nerve number 7, that's facial nerve, and even also what we call the glossopharyngeal nerve, are going to be taking the sense of the taste, the central nervous system, to the different cortex within the cerebrum. From there, the information will come down to what we call the hypothalamus, specifically on what we call the parasympathetic portion of the hypothalamus, which will give off the descending axon to the medulla. From there, the motor fibers of the vagus nerve will come off through the jugular filament, will come down to the gastric glands and release acetylcholine, which will bind on M3 receptors, leading into what we call a stimulatory effect to the production of the gastric juice. Same thing is going to be happening for the case of the smell, same thing for the case of the thought, same thing for the case of the frying sound. Different cortex will be activated, which will give off the information to the hypothalamus, specifically the parasympathetic part, which will also give descending axon to activate what we call the dorsal nucleus vagus, which will give out the motor fibers of the vagus nerve to go and release acetylcholine on different cells within the gastric gland, leading to the production of what we call the gastric juice. Okay, so now that's generally how the cephalic phase is going to be happening. And the main inhibitors of the cephalic phase is going to be anything that's going to be activating sympathetic nervous system. Okay, for example, we have what we call stress. Whenever you are stressed, sympathetic nervous system is going to be taking over. Even also what we call the fear. Okay, and even also any kind of the anxiety, any kind of the emotions, anything that's going to be activating sympathetic nervous system will inhibit all the effects of what we call the vagus nerve. Okay, and then another thing is that the bad smell. That one is also enough to inhibit gastric secretions. Okay. That's a little summary of what we call the cephalic phase. When we come to the gastric phase, this is the second phase of the gastric secretions. This one is going to be accounting for 2/3 of the gastric juice within the stomach. It is going to be leading to the production of a lot and a lot of the gastric juice. I'll be taking much little of the time to talk about this gastric phase because it is accounting the biggest percentage of the gastric juice within what we call the stomach. Now, in this kind of the phase, we are going to be having food already inserted into the stomach. Okay. In the previous phase, which was the cephalic phase, there was no food in the stomach. But within the gastric phase, there is food in the stomach. Now, what is going to be also a driving factor, which cranial nerve is also going to be driving this kind of this phase. We're also going to be having vagus nerve effect. We are going to be seeing how the vagus nerve is coming about. Now, what are the main stimuli for gastric secretion during the gastric phase? The first one, which is super very important, is what we call the distension, or what we call the stretch. Whenever food enters into the stomach, there's that kind of the stretch. Okay. Now that kind of the stretch, we have what we call stretch receptors, and these stretch receptors are coupled with what we call nerves, and these nerves are sensory nerves of what we call cranial nerve 10, and that's what we call the vagus nerve. Now, the vagus nerve will be taking the sensation of the stretch to the central nervous system, primarily what we call to the medulla oblongata. From there, we shall have activation of what we call the dorsal nucleus vagus, which will give off activated motor fibers that will come over through and pass through what we call the jugular foramen down to what we call to the stomach and release acetylcholine on different cells, like for example the parietal cell, the chief cell, the enterochromaffin-like cell, to produce their own kind of the secretions. It releases acetylcholine on enterochromaffin-like cells. It will stimulate the enterochromaffin-like cell to release what we call histamine, of which histamine can stimulate the parietal cell to produce what we call hydrochloric acid. Okay, such a beautiful thing. So now there is stretch, which is going to be among the main stimuli of gastric phase. Another one is going to be what we call the partially digested proteins. And when we talk about the partially digested proteins, a good what to use is what we call the peptones. Okay, what happens is that within the antrum of the stomach, we have those kind of specific type of cells that are sensitive to what we call partially digested proteins, which we call the peptones. And these cells are what we call the antro-G cells. When these G cells are stimulated by what we call the peptones, they are going to be releasing a molecule which we call gastrin, and of which gastrin can all move into the blood and goes and stimulate different cells within the gastric gland, the likes of the chief cell, the likes of the parietal cell, and stimulate them to produce their own kind of the secretions. Okay. This is also such another beautiful thing. So now, peptones is also one of the main stimuli of gastric secretion during the gastric phase. Okay. And now, when we talk of the main inhibitors of gastric secretions during the gastric phase, we have the first one, which is super very dangerous, is what we call the somatostatin. Now, somatostatin is going to be coming from what we call the D cell. Now, how is somatostatin getting produced? Now, somatostatin is produced by what we call the D cell. Now, these D cells are super much sensitive to what we call high levels of hydrochloric acid. When the stomach becomes super concentrated with hydrochloric acid, these D cells, they become activated. They have chemoreceptors that sense high levels of hydrochloric acid. Well, what happens is that when these D cells are stimulated, they will release a molecule called somatostatin. This somatostatin can all move in blood and goes and inhibit the G cell, the antro-G cells, from producing gastrin. If you inhibit the antro-G cell from producing gastrin, it means you're going to be having no gastrin, and of which this one will also lead into no stimulation of what we call the parietal cell and then the chief cell from producing their own secretions. Okay, so now somatostatin can inhibit the antro-G cells from producing gastrin, of which gastrin is a powerful stimulator of what we call the chief cells and then the parietal cell from producing their own secretions. It sounds more beautiful. Okay. Besides that, somatostatin can all move direct to the parietal cell and inhibit the parietal cell from producing hydrochloric acid. Okay. Such another beautiful thing. So now we have another kind of the main inhibitor is what we call anything that activates sympathetic nervous system will automatically block the parasympathetic effect, what we call gastric secretion, by what we call the vagus nerve. So now what we call stress, depression, and fear, all those ones can inhibit gastric secretion even during this phase. Okay. And then also have what we call the parietal cell stimulators. This parietal cell is going to be stimulated by different substances. The first one is what we call acetylcholine. from the vagus nerve. Acetylcholine can stimulate the parietal cell, leading to production of what we call hydrochloric acid. You should not forget, you also have what we call intrinsic factor coming from what we call the parietal cell that aids in what we call absorption of vitamin B12. We also have what we call gastrin also stimulating the parietal cell. We also have what we call histamine binding on H2 receptors, which can also stimulate the parietal cell to produce hydrochloric acid. Okay, and then also besides being stimulated, this parietal cell can also be inhibited by different substances. For example, somatostatin can inhibit the parietal cell from producing hydrochloric acid, and even also another kind of the molecule which we call prostaglandins E2. Prostaglandins E2 can bind on EP3 receptors which are residing on what we call the parietal cell and inhibit the parietal cell from producing hydrochloric acid. It is such another beautiful thing, okay? So now that summarizes the gastric phase of gastric secretions in the simplest way possible. Okay, so now having seen the cephalic phase, the gastric phase, let's go ahead and look at what we call the intestinal phase of gastric secretions. Okay, now the key most important thing with the intestinal phase is that the kind of the gastric juice which is going to be produced during this phase is going to be very small. It doesn't account for a significant amount of the gastric juice during this phase. Okay, now what is going to be significant for this phase? The main purpose of this phase is going to be aiding at inhibiting gastric secretions and even also inhibiting what you call the stomach motility. The whole point is we want to limit how much of the chyme goes to the intestine such that we give the intestine, the small intestine, enough time, a good medium that will help to finalize the process of chemical digestion. If we allow more of the chyme to leave the stomach into the duodenum, that one, remember, the chyme is going to be moving with a lot of different substances. Now, let's first look and see how is this chyme going to be leaving what we call the stomach. Now, the stomach is designed with a lot of layers, of which one of these layers is going to be a muscular layer, which we call external muscularis. Okay. Now, that layer is going to be causing a peristaltic wave, which is going to be a wave of contraction that will contract from the fundus down to what we call the corpus, down to what we call the pyloric antrum, and then on what we call the pylorus. We have a strongest peristaltic wave on what we call the pylorus. Now, when that one contracts strongly, it will squeeze some of the chyme to move from the stomach into what we call the first segment of the small intestine, which we call the duodenum. However, a biggest percentage is going to be pushed back to the fundus or even what we call to the body for proper chemical and what we call mechanical digestion. And this mechanism of the stomach contraction to push back some of the chyme to the fundus and then to what we call the body for proper chemical digestion is what we call a retropulsion. Okay? Now, some of the chyme, approximately to 2 to 3 milliliters, is going to be entering what we call the first segment of what we call the small intestine, which we call the duodenum. Now, that chyme is going to be containing a lot of different substances that are going to be moving with that chyme. Some of those substances are going to be having what we call hydrochloric acid. So now this chyme, which is going to be leaving the stomach, is going to be moving with a lot of the acids. We don't want a lot of these acids in the duodenum, which will lead into what we call duodenal ulcers. But the duodenum is going to be having those kind of the mechanism that are going to be aiming at reducing this kind, at neutralizing this kind of the protons to make sure that the duodenum is free from acidic medium, which will also favor what we call enzymatic action, what we call from the pancreatic juice. Okay, so now this chyme is going to be having a lot of the protons, going to be having partially digested proteins, which we call the peptones. It's going to be having what we call the fatty acids, or generally the fats, and even also short chains of what we call the carbohydrates, which we may call the oligosaccharides. Okay, so now these are the molecules which are going to be moving along with what we call the chyme. Now, what happens is that these kind of the molecules are the ones which are going to be causing a lot of the stimulation of different cells, of which these cells will release different chemicals that will either stimulate the gastric secretion or inhibit gastric secretions. Before I forget that this phase or the intestinal phase of gastric secretion is going to be happening whenever chyme enters into what we call the small intestine. It sounds more beautiful, okay? So now we're going to be having now chyme having a lot of different substances I've just talked about. They're going to be stimulating a lot of different cells within the mucosa of what we call the small intestine. Now, we're going to be having these peptones. These peptones are going to be acting on what we call the intestinal G cell. What happens is that when peptones act on intestinal G cell, it will lead into the production of what we call gastrin. Gastrin is a hormone which can flow into the blood, and then it goes and stimulates the parietal cell to produce hydrochloric acid. So now, peptone's effect on intestinal G cell is going to be one of the main stimuli of gastric secretions during the what we call the intestinal phase. And another kind of the stimuli is what we call enteroxytocin. Enteroxytocin is one of these important molecules which is produced by some of the endocrine cells, of which their names are not yet so much identified, but also this enteroxytocin molecule can move into the blood and also go to the parietal cell and stimulate the parietal cell to produce hydrochloric acid. Okay, such another beautiful thing. Now, the proceeding effect is going to be what we call the inhibitory factors to the production of what we call the gastric juice. Let's go step by step and see how gastric secretion is going to be inhibited during the intestinal phase of what we call gastric secretion. So now, the inhibitory effect is going to be exerting the biggest effect in the production of what we call gastric secretion. So now, let's go ahead and see it step by step and see how this one is going to be happening. Now, within the small intestine, particularly what we call the duodenum, we have specific type of cells. And these cells are what we call the S cells. These S cells, they are sensitive to what we call fatty acids, which are in chyme. They are also sensitive to what we call the protons. What happens is that when these S cells are stimulated, they are going to be releasing a hormone which we call secretin. Secretin can move into the blood, and it goes and inhibit what we call the G cells from producing what we call gastrin. If you inhibit gastrin, it means you're inhibiting stimulators of parietal cell from producing hydrochloric acid. Such a beautiful thing. So now another thing is that a secretin can also move into the blood, and it goes to the liver and stimulates the liver to enhance the production of what we call bile. Why? Bile is going to be very important in emulsification of what we call the fats. It sounds more beautiful. Okay? And then another thing is that a secretin can also come over to the pancreas, specifically on the cells which we call the ductal epithelial cells. Stimulate the ductal epithelial cells, leading to the production of what we call bicarbonate. Bicarbonate will come over to the duodenum and neutralize what we call the protons, leading to the formation of what we call carbonic acid, which will eventually break into what we call water and what we call carbon dioxide gas. The carbon dioxide gas is going to be exhaled during ventilation, and then water will be used to run some other kind of the processes during the intestinal, what we call motility within the GIT. Okay, secretin is such another beautiful thing. We also have some other kind of cells which we call the eye cells. These eye cells are sensitive to what we call peptones. They are also sensitive to what we call the fatty acids, and even also what we call oligosaccharides. What happens is that these kind of the molecules are going to be stimulating the eye cell, leading to the production of what we call cholecystokinin. Now, cholecystokinin is super very important. Cholecystokinin will move into the blood. It goes and inhibit the parietal cell from producing hydrochloric acid. It can also exert a minor effect on what we call the chief cells from producing their own kind of the secretion, the likes of pepsinogen, and even also what we call the gastric lipase. Besides that, cholecystokinin can also move into the liver and enhance the process of bile formation because it is sensitive to what we call the fatty acids. So we may need what we call the bile in emulsification process, which is going to be happening in what we call the duodenum. It sounds more beautiful. Another thing is that cholecystokinin can also move into what we call the gallbladder and stimulate the gallbladder contraction, which will lead into ejection of what we call the bile into the first segment of the small intestine for proper what we call emulsification of fats. We also have receptors of what we call cholecystokinin or what we call the sphincter of Oddi, which will make it relax, allow the bile to enter into what we call the duodenum. All of it makes a lot of sense. Okay? Besides that, cholecystokinin can also move into what we call the pancreas and then stimulate what we call the pancreatic acinar cells. Once these pancreatic acinar cells are stimulated, they will start releasing a lot of the pancreatic juice, of which these pancreatic juice are going to be containing a lot of enzymes: pancreatic lipase, pancreatic amylase, pancreatic nucleases, pancreatic proteases. All those ones will come over to the duodenum and start the process of final chemical digestion of different food substances. It makes a lot of sense. Okay? And then we also have some other kind of the cells within the duodenum, and these cells are what we call the K cells. These K cells are sensitive to what we call fatty acids. They are also sensitive to what we call oligosaccharides. What happens that when these K cells are stimulated, they are going to be releasing a molecule which we call gastric inhibitory peptide, which some other people call glucose-dependent insulinotropic peptide. Okay, so now what happens is that this molecule can also move to the blood. It goes to what we call the parietal cell, and it binds on specific receptors on the parietal cell and inhibit the parietal cell from producing hydrochloric acid. Okay, so now you are seeing that secretin, CCK, gastric inhibitory peptide are all exerting an inhibitory effect to the production of what we call the gastric juice. Besides that, gastric inhibitory peptide can all move to what we call to the pancreas, and specifically it go to the simplest kind of the structure which accounts for 1% of the tissue that make up the pancreas. You know how that kind of the tissue is called? That tissue is what we call the islets of the Langerhans. It has a multiple of different kind of the cells, but the cells of interest in this case are what we call the beta cells of the islets of the Langerhans. What happens is that gastric inhibitory peptide will come over on those beta cells and stimulate the beta cell to produce what we call insulin, of which insulin has a positive influence in the duration of what we call lowering what we call the glucose levels. Besides that, insulin has another kind of a metabolic role of what we call glycogenesis, what we call cellular glucose uptake, lipogenesis, and even also protein synthesis. It makes a lot of sense. Okay, and then another kind of the inhibitor, and of course we said that anything that activates sympathetic nervous system will automatically inhibit what we call gastric secretions. Okay, it makes a lot of sense. So now Umunad, we have talked a lot of things, and if you think whatever I have talked is making sense, is adding to you, you are benefiting, please, you can support it, and that one can be done by a couple of means. You just hit the like button, comment down in the comment section. You can share this video with someone else who you think that this video makes a lot of sense, and of course, don't forget to subscribe at the end of it all. Thank you very much for watching Tumunad as always. Until we meet next time.